Good morning, one and all. I am Himavashni here with our topic called as RNA and extraction of RNA. Acha, let us know about the RNA. RNA is the ribonucleic acid is an important biological macromolecule that is present in all the biological cells. It is principally involved in the synthesis of proteins carrying the messenger instructions from the DNA, which itself contains the genetic instructions required for the development and maintenance of the life. There are three types of RNA. Of many types, out of many types we know of RNA, we are well known with three. There are messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. So these are present in all the cells, all the organisms. Generally where the RNA is found and DNA is found, there are two types of nucleic acids which are the polymers found in all the living cells. That is DNA and RNA. DNA, deoxyribonucleic acid, is mainly found in the nucleus of the cell. RNA, the ribonucleic acid, is mainly found in the cytoplasm of the cell, although it is usually synthesized in the nucleus. Functions of the RNA. The central dogma of the micromolecular biology suggests that the primary role of RNA is to convert the information stored in DNA into proteins. In reality, there is also much more story to the RNA store. The other functions of the RNA. The messenger RNA or the mRNA serves as the temporary copies of the information found in the DNA. Ribosomal RNA or the rRNA serves as the structural components of the protein making structures known as the ribosomes. tRNA or the transfer RNA carry amino acids to the ribosomes to be assembled. About the structure of the RNA. Generally, in the structure, the DNA is of double helical and double stranded, but the RNA is of single stranded. Ribonucleic acid is a molecule similar to the DNA. Unlike DNA, the variation is RNA has a single stranded. It is made of alternating sugar that are ribose and also phosphate groups. This is the sugar phosphate backbone. Attached to the sugar is one of the four bases. They may be either of adenine abbreviated as A, uracil abbreviated as U, cytosine abbreviated as C, or the guanine abbreviated as G. Let us know about the differences between the RNA and DNA. DNA is known as deoxyribonucleic acid. RNA is known as ribonucleic acid. DNA, two strands in the double helical formation. RNA has a single strand. DNA, it generally stores the genetic information, while the RNA, it builds the protein from the genetic information. DNA contains the slightly different sugar deoxyribose, and RNA has a nucleobase uracil, while DNA contains a thymine. Coming to the nucleotide base pairs of the DNA, it contains cytosine, guanine, and adenine and thymine. Nucleotide base pair of RNA are cytosine and guanine, adenine and uracil. In the difference between DNA and RNA base pairs are thymine is replaced by uracil in RNA. Let us know about the extraction of RNA. I am here going to explain about the different topics of extraction extraction of RNA are: what is the extraction of RNA? What is the purpose of extraction of RNA? What is RNA extraction kit? And what is the role of chrysol in extraction? What is RNA extraction? RNA extraction is the purification of RNA from biological sample. The procedure is complicated by the ubiquitous presence of ribonuclease enzymes in cells and which can rapidly degrade the RNA. What is the purpose of extracting the RNA? RNA extraction is the purification of RNA as per we know before. The procedure is complicated by the ubiquitous presence of ribonuclease enzymes in the cells which can rapidly degrade the RNA. That is, which rapidly degrades the RNA is the purpose of extracting. What is the role of chrysol in RNA extraction? Chrysol reagent is an acid vanadium phenol based reagent ideally designed for the extraction of RNA as well as DNA and protein from various biological inputs, sample input. The low pH of that is the acidic of trisol controls to separate RNA from DNA and protein, while a high pH can cause RNA and DNA to be isolated together. What is RNA extraction kit? The RNA extraction kit enables the total RNA extraction from tissues. The extraction and homogenization of tissue samples can be carried out rapidly and efficiently using the biorupture. As sonication medium maintains the integrity of RNA while disrupting cells and dissolving the cell components. Let us know the extraction of RNA by an experiment. Let us learn the extraction of RNA from liver sample by a Hot-Phenol Method experiment. The objective of the experiment is to extract the RNA from liver sample by Hot-Phenol Method. The principle of the extraction of RNA of liver cells by Hot-Phenol Method is the tissue or cell sample is homogenized or disrupted in hot phenol. Chloroform is mixed with the lysate and the mixture is separated into three phases by centrifugation. The RNA is then precipitated from the aqueous phase with chilled ethanol. The Hot-Phenol method is to produce high yield of the RNA. Let us know about the preparation of the extraction buffer. To prepare 100 ml of extraction buffer, we need to take initially 5 ml of 2M trihcl and 0.5 ml of EDTA of 4 ml is added and 1.4 ml of NaCl of 2.1 grams is added and 0.5% of SDS of 0.5 grams volume and finally 0.2% DDT of 200 microliters is taken and made up to 100 ml with distilled water. Let us know about the materials required for the experiment. The materials required are extraction buffer, as we have seen in the previous slide how the extraction buffer is prepared, and DEPC, chloroform, isoamyl alcohol, ethanol and phenol are taken. Let us know how the procedure of the experiment is to be worked out. Initially, 50 mg of liver tissue is taken and homogenized at 3000 rpm for 3 to 5 minutes to collect the tissue cells. And 400 microliters of extraction buffer is added to the 400 microliters of homogeneous sample and they are placed at 100 degrees centigrade for 5 minutes with occasional stirring or the mixing. Then the sample is transferred directly into pre-warmed alkyl acid phenol is to chloroform, which are in the ratio 5:1, and mix the sample by vortexing at a high speed. Then the sample is placed at 65 degrees centigrade for 10 minutes with a periodic mixing. Then spin the samples at 15,000 rpm for 5 minutes and collect the upper aqueous layer into a fresh vial. That is, the samples are spinned at 15,000 rpm for 5 minutes and the upper aqueous layer is carefully taken into a fresh vial. And an equal volume of pre-warmed acid phenol is to chloroform are added at a high speed. Then again spin the samples at 15,000 rpm for 5 minutes and collect the upper aqueous layer again into another fresh vial. Add equal volume of chloroform is to isoamyl alcohol, which are in the ratio 24:1, and mix the sample by vortexing at high speed. Spin the samples at 15,000 rpm for 5 minutes and collect the upper aqueous layer into another fresh vial. Add equal volume of chilled ethanol and place the sample at minus 20 degrees centigrade for 10 to 15 minutes. Then spin down the RNA by centrifuging at 12,500 rpm for 20 minutes. Then discard the supernatant and air the pellet at the room temperature. And add 50 microliter of TA buffer into the pellet and mix it well. Now, as finally, we should check the purity of the RNA by taking the OD values at 260 by 280 in a spectrophotometer and analyze the sample on 1% agarose gel. After doing the result of the experiment we get is, we get the absorbance ratio of RNA sample at 260 by 280 is greater than 2.0. The greater than 2.0 absorbance ratio explains us the RNA.